All right, we're getting into the last sections here of this massive, right, PowerPoint. So before we start this last concept, I guess we can say, is that there's a couple important terms or terminology to go through as we move forward. So we think about two neurons in a sequence. Remember, neurons have polarity, meaning information travels in one direction, right? From dendrites to cell body, that's graded potentials. If it's strong enough, we get an action potential from the axon hillock all the way to the axon terminal. When we get to the axon terminal, neurotransmitter is released. Depending on what part of the nervous system we're in will depend on the neurotransmitter that's released, whether it's acetylcholine, epinephrine, norepinephrine. We'll worry about those a little later. But the neurotransmitter will then have an effect on the next cell. This cell happens to be another neuron, so the dendrites here will receive the neurotransmitter, and you either get depolarization or hyperpolarization, right? And that would be a graded potential that would move down this dendrite, these dendrites in this cell body, that's strong enough to produce an action potential, right? So you get this relay, basically, is the point from neuron number 1 to neuron number 2. And that relay of information right here is the synapse. So we have different names for these two neurons that occur before the synapse or after the synapse. So we'll have a presynaptic neuron. This is our presynaptic neuron. It will send the information in the form of a neurotransmitter, like a baton in a relay, to the postsynaptic neuron, and the postsynaptic neuron receives the information from the presynaptic neuron. So pre and postsynaptic neurons will be two important terms moving on to the kind of final set of slides in this chapter. What's happening at the synapse? This is information you likely already know if you studied contraction of skeletal muscle, where a neuron stimulates the skeletal muscle. You might remember the acetylcholine has to fly across the gap at the and bind to the motor end plate on the muscle fiber. Right, that's all part of this exam as well. But in this case, we have a presynaptic neuron and a postsynaptic neuron instead. So the process is the same. The action potential will arrive at the axon terminal of the presynaptic neuron. Calcium happens to rush in at that point, which don't worry about this big word in bold here, but that causes vesicles to release through exocytosis neurotransmitters into that synaptic gap or cleft, which binds to the postsynaptic neuron. And continuing the story, so that presynaptic neuron released the neurotransmitter diffuses across the synapse, and I jumped the gun on the previous slide, but it binds to a receptor on the postsynaptic neuron. That opens up a channel, and since it's a neurotransmitter that caused a channel to open, it's a chemically gated channel. The ion channels then open, and you have sodium rushing in, potassium rushing out. We have all these ions just kind of rushing across the membrane with these channels open based on their concentration gradients, and that causes a graded potential. Now, what's new here is a graded potential could be excitatory or inhibitory. So now we need to go through those scenarios. Right? This is graded potential, so we're at a dendrite. And just think of that picture of the presynaptic neuron and the postsynaptic neuron, where that relay is, where that synapse is. It's at the dendrite. So remember, at dendrites we have graded potentials. So graded potentials can be excitatory or inhibitory, and we'll kind of move on from that story with with the next slide. Actually, before we move on, a couple points about the neurotransmitters. So the neurotransmitter bridges the gap in the synapse, right? The presynaptic neuron releases a neurotransmitter, and the postsynaptic neuron receives it. But their effects are degraded or taken away right away. And it says within a few milliseconds, neurotransmitter effect is terminated. And these are the three ways that neurotransmitters will only have an effect for a very short period of time before they're taken away. I'll explain these three on the picture on the next slide. So here's the axon terminal of the presynaptic neuron, and there's kind of the dendrite of the postsynaptic neuron. So there's your synapse. there's your exocytosis of neurotransmitters. So the neurotransmitter flies across the synaptic cleft. We'll get bigger here. And then it's going to bind to a receptor on the postsynaptic neuron. That's going to cause a depolarization or a hyperpolarization on this graded potential. But the point of this slide here is when the neurotransmitter binds, they're not showing a bound here. They're showing one bound right here. So I'll use this picture. When the neurotransmitter binds to this receptor protein and has its effect, we don't want that effect to last. For example, if that mosquito lands on your arm and then flies away, we don't want to keep feeling that mosquito on your arm forever because it's gone, right? We only want it to last as long as it needs to last. And when the stimulus goes away, we want our sensation to go away. So how do we get rid of this neurotransmitter? Well, there's three ways. So neurotransmitter, once it binds, often it gets taken right back by the cell that released it, by that presynaptic neuron. That's called reuptake. Just like it performed exocytosis here, well, it performs endocytosis, right, to take it back. Also, these neurotransmitters that are in that synaptic cleft get broken down by enzymes. So enzymes come in and just break them up so they don't just diffuse across and bind because the event is already over. And some of them just kind of diffuse away, right? So neurotransmitter gets released and some of them just kind of escape. So there's kind of three ways that neurotransmitters, although they have their effect momentarily, we need that effect to go away just as fast as it came. So we're going to put all of this together now. And it doesn't say, but we're talking about graded potentials here, because when you have a presynaptic neuron forming a synapse with a postsynaptic neuron, well, at the synapse, it's the dendrites, could be the cell body too sometimes, but let's just stick with the dendrites that are receiving their neurotransmitter, right, from the synapse. And it's dendrites that produce graded potentials, not action potentials. Where does the action potential begin? Well, right here, right at the axon hillock, and it travels down the axon. So when we're talking about the graded potential that occurs at the postsynaptic neuron. So neurotransmitter is released by the presynaptic neuron and binds to the postsynaptic neuron. And we said this graded potential that weakens with distance, graded potential could be excitatory or inhibitory. So here's our big term here for excitatory and inhibitory, EPSP and IPSP. You can have an excitatory postsynaptic potential or an inhibitory postsynaptic potential. Excitatory means, yeah, it's going to be more likely that we get an action potential. Inhibitory, it's going to be less likely. So let's explain these two here as we move to the. So what happens in an excitatory postsynaptic potential? Let's read through the text, at least the important part, and then we'll look at the picture on the next slide. So an excitatory synapse, basically sodium rushing in is greater than potassium rushes out. So the effect is depolarization. And remember, depolarization on the graph raises it from -70 closer to 0. And if we're getting closer to 0, depolarization, that means we're more likely to get an action potential if we're at threshold at the axon hillock. So an excitatory synapse, an excitatory postsynaptic potential, sodium going in is greater than potassium rushing out, resulting in depolarization. So let's look at the picture. So this is looking at the dendrite of that postsynaptic neuron, here we have the stimulus, and we saw the stimulus brought it closer to that positive. And by the way, once it gets to threshold, -55, that means it's going to go all the way to a +30. So we don't have to get all the way up there and depolarization to produce an action potential. Threshold means it's going to happen automatically. It's going to shoot up at that point. So we're really just looking to move up 15, -70 to -55. But that's fine. In this case, yeah, we had a stimulus. It caused us to get closer to threshold. So that's depolarization. So it's excitatory, an excitatory postsynaptic potential, which you can see down here. Just the opposite, we can have inhibitory postsynaptic potentials. And in this case, it doesn't say it, but now potassium moving out is greater than sodium rushing in. But the point here is that we get hyperpolarization of the cell, making an action potential less likely. So let's look at this on the picture on the next slide. So here's our resting membrane potential, and our dendrite received the neurotransmitter, but this happened to cause depolarization. This happened to cause depolarization, and if we look, now we're farther from threshold than we were at rest. Remember, -70 is your resting membrane potential, and since we moved farther from threshold, now it's less likely that an action potential will occur because it has much farther to go than it did originally. So this type of stimulus is inhibitory, so it's an IPSP, an inhibitory postsynaptic potential. Right, and always remember it's hyperpolarization that causes the inhibitory postsynaptic potential. And what we already saw before, it's depolarization that causes excitatory postsynaptic potentials. And what we're going to see on the next few slides here is a concept called summation. So here's an important point that we have yet to bring up, that we will in the last part of this PowerPoint, is that one single, so just one EPSP, and that's a graded potential, but it's an excitatory graded potential. We call it an excitatory postsynaptic potential. A single one has no chance of producing an action potential, right? So even though it's excitatory, yeah, it's not going to get to the axon hillock because this is graded potential, meaning it's going to lose, even though it's excitatory, it's going to lose steam as it travels towards the axon hillock. So yeah, one single little excitatory postsynaptic potential from one little stimulus is not going to cause an action potential. What you have to do is take a bunch of these excitatory postsynaptic potentials, add them together, and adding is summing, right? So that's called summation. And that summation has to reach threshold at that axon hillock. So yeah, there's a lot of words here, but let's go to the next slide and then to the pictures, and we'll be able to kind of put this all together.